need a call last week.
Hey, we're going to get started with the next session where everybody's mic'd up and everybody's
Up to you.
online. And want to go ahead and bring up the slides for this session real quickly? No, the intro slides. Oh, clicker? Okay. Magic. Okay. Okay. Can you hear us, Shubhanji and Amy? Okay. Just we'll wait a second, everybody. We're gonna start. Just to orient people to this conversation for you who aren't living and breathing like some of us on the stage. This is sort of a bit of an update. We're going to talk about this framework as for data acquisition and sharing and using data most efficiently. For you that aren't familiar, the Act for ALS was signed, which really was the nidus for many programs. Private partnership. In part two, there were things related to grants and expanded access programs. But this was really an act that really was to reinvigorate and to stimulate research in ALS. And so what this has done is create a lot of different programs. And there's three that we're going to highlight today. And we'll talk about the organizations that align with them. And really as a model that we can discuss and actually address some of the things that we talked about in the last session. So critical path is involved in the critical path of rare neurodegenerative diseases. And obviously, real world data is important to us, but with really adding, our expertises with the solutions on taking clinically aligned data and existing data in particular to really identify and to develop things like, biomarkers and trial simulation tools and other solutions to advance drug development. Obviously, our world is in existing data, but also understanding where the gaps are, where are the holes. The next program that's really important, and Amy will talk to this, is the ALS effort, which is sort of the what I call the testing lab. It's a network of sites that she'll elaborate a little more on, but really it focuses on, it's a large natural history study that serves as a bit of a resource, as well as, from a classical perspective of just a normal natural history study, but a place where those gaps can be filled. And then the middle one is AMP-ALS, which is an FNH initiative that really is focusing on funding and developing programs to help design and implement within the ALS effort. And we'll talk about those, and we'll get deeper dive in those discussions and the questions. Now, what's really nice about this is that, as I said, this is a nice incubated system. It's really a nice space. This is a modification of sort of the learn and confirm paradigm, for those of you who might be aware of it, where we have different partners all contributing to a whole to really advance drug development and to understand the natural history and disease, all leading to the efficient study and execution of the discovery of new therapeutics. So like with us, we align with FDA and the regulatory strategy, producing drug development tools through CPR&D. AMP-ALS is with FINH and NIH and its members, really to help fuel and develop new science to fill those gaps. And then the All-ALS effort was really led by NIH, is really a space where we work to deploy and understand the natural history study, but also things to test new hypotheses and to develop things like new biomarkers or new COAs and whatnot. Working all together, we work with our partners, both at FDA and NIH, to really provide oversight and input as we work through this. And I even if Teresa wasn't here, they've heard me say, Teresa, Michelle, and others, they spend a lot of time and energy with us in this initiative, really, providing their counsel and their advice and troubleshooting very complicated situations where it would help fuel the innovation and to drive the science. And centered to all this is that there's a group of individuals who give us generously of their time, and that's persons with lived experience. We're very fortunate with the community to sit on the different working groups and to be involved in multiple aspects and providing advice throughout the different working groups that are being discussed, but also to keep us mindful of what we're doing and the timeliness of that and doing this. And so what I'm going to do is start introductions with folks, And we'll start into the questions. We can start online first. And Amy, do you want to start with yourself? And Shubhangi, do you want to go next?
Sure. Good morning, everybody. My name is Amy Zhou. I am a program director in the Division of Clinical Research at the National Institutes of Neurological Disorders and Stroke, or NINDS, and the lead for the All ALS effort, and really grateful to be here today and part of this conversation.
And Shubhangi, do you want to go ahead and introduce yourself?
Yes, sure. Hi, I'm Shubhangi Lal, the project manager for the Accelerating Medicines Partnership Program in ALS at the Foundation for the National Institutes of Health, or FNIH. I'm really excited to be here and talk more about AMP-ALS.
Okay. And just to start, since we're on the panel online, like Amy, do you want to elaborate, more what's going on in all ALS and maybe, what kind of the data is being generated and contributed or planned to be contributed to this ecosystem? And one of the questions that was discussed earlier, how you're addressing in particular the issues of diversity and inclusion, because that was one of the really important factors that was involved in getting data from a very diverse population across the United States.
Yeah, sure. I'm happy to kick off, and I'm always excited to talk about this really important initiative. So All ALS is a large clinical research consortium that's been funded by the NIH. All ALS stands for Access for All in ALS. And the goal of the consortium, as Collin already alluded to, is really to support the AMP-ALS by collecting the data and samples necessary to advance their research goals. And, one of these goals is really to help develop better biomarkers for diagnosis and monitoring of disease. And to support that goal, All ALS is conducting a large natural history study to collect high-quality, clinically annotated longitudinal biospecimens, specifically blood and CSF. Now, how are we going to do that? So I'm just going to say a few words about the structure of the consortium. So it's a big consortium. At this point, we have 35 clinical sites. They are spread across 29 states and Puerto Rico. And the goal is to enroll 3 different cohorts of individuals. So first, individuals that have ALS. Secondly, individuals that are at high risk of developing ALS because they're a gene carrier for a particular mutation, but they don't have any symptoms yet. And the third cohort is a control cohort. So the goal is to follow these cohorts for 2 years and with regular check-ins at regular intervals. And, all of the data that is collected from this is going to go into a knowledge portal that we hope, that the plan is to make that data freely available to researchers and the public. Now, regarding this question of, really making sure that we are capturing a representative sample of individuals with ALS, the sites for All ALS are, a majority of those are located in areas with a high representation of these underrepresented populations. And, that was deliberate. And our hope is really that that will make this consortium well poised to really capture these populations. Another thing I just want to mention is that one of the things that we're really excited about is that All ALS is going to be able to enroll participants remotely. And, it's possible that this will allow individuals that don't live close to a large academic center to be able to participate in this study. So we are really excited about this effort. So this award was made actually just only about a year ago, and we're really thrilled to say that the study actually began enrolling last month and has already enrolled 20 participants. The goal is an ambitious one. The goal is to enroll over 2,000 participants by early 2026.
Thank you, Amy. Shubhangi, do you want to relay what AMP programs are, and then maybe the work streams that we're working diligently on?
Sure. So before I get into what is an AMP program, I'll just take a minute to talk about FNIH. For people who don't know about it, FNIH is an independent, non-profit organization that is dedicated to advancing biomedical research. FNIH convenes and manages public and private partnerships between NIH, academic institutions, private partners, and patient advocacy groups. This is to accelerate drug development. One of FNIH's flagship initiatives is the Accelerating Medicines Partnership Program, or the AMP program. The AMP program leverages public and private resources to enhance our understanding of disease pathways and accelerate drug development. So we bring this about by consensus between the public sector, NIH institutions, FDA, and the private players like pharmaceutical companies and biotech companies, along with patient research organizations. The AMP programs operate in a pre-competitive environment, and they promote data democratization. To date, FNIH has convened 12 AMP program, and this is in various disease areas across neuro, cardiac, and immunity and inflammation. The newest AMP project that was launched in May of this year is the AMP ALS project. This is, as an, you just heard Collin talk a little bit about Act for ALS, and as a component of the Act for ALS, AMP ALS was set up to bring together NIH, FDA, Critical Path Institute, and several other private partners, including pharmaceutical companies and patient organizations. So talking a little bit about the structure of AMP ALS, at the top, we have a steering committee. This constitutes of representatives from the public and private partners of AMP ALS. This is the decision-making body within AMP ALS. This basically decides the direction the program will move into. The steering committee works in close collaboration with the working groups. And the working groups are where the granular discussions take place based on the research areas that have been identified by the program. So to start off with, as Collin mentioned, at the beginning, at the launch of the AMP ALS project design phase itself, the first working group was set up to include to include the people with lived experiences. We want to make sure that we are able to bring all aspects. We can bring in their involvement in all aspects of the AMP ALS project. This group includes those not just diagnosed with ALS, but also gene carriers, caregivers, and this group, members from this group are also a part of our technical working groups. At this time, we have 4 technical working groups. We have a data portal working group, which will refine the data architecture framework. Just to provide a background, one of the focus areas of AMP ALS is to set up, establish a knowledge platform. This will be a one-stop shop for researchers to access existing data sets. This will also include data that will be generated from the All ALS study, as Amy mentioned. So this working group will refine the data architecture framework, oversee the establishment of the ALS knowledge portal, procure existing data sets, and develop data analytical tools for it. The second working group that we have currently is the biomarker working group. This will prioritize and drive the implementation of the key biomarker research objectives that have been outlined. The focus of this group will be to validate promising assay candidates. The next working group that we are working on is the natural history study working group, which will guide the research efforts within the All ALS consortium. finally, we have at this time the clinical endpoint working group. This group is being managed by the Critical Path Institute. The emphasis of this group is to optimize existing clinical outcome assessments and develop new ones. This will be done by conducting a COA landscape analysis, generating psychometric data for new and modified COAs, and will be assembling evidence to support COA selection. So these are the working streams that we currently have, and these are dynamic in nature. And these are not permanent, and depending on the need of the project, more such groups will be formed. So these are the groups that we are currently working on.
Thank you. In addition, I mean, y'all have heard some of the conversations today, and not today, but yesterday around modeling and simulation. As we aggregate data into the warehouse, what will end up happening is there'll be a group that focuses on the biological disease staging, but also then, modeling and simulation and developing of other tools that might be applicable in this space. I want to switch gears and connect with Ramona. Ramona, you've been in this regularly and participating in these working groups. One of the things I really want to discuss, because for those of you that know, the data warehouse will really be made of two separate components that all come into a federated model. So from the data request to the warehouse will appear as one. But one of the cornerstones of the data warehouse will be RDCA DAP, which you've seen discussed in other sessions. And so, Ramona, do you want to explain how you feel the importance of the data sharing, particularly some of the areas that you're interested in, because obviously you're very adept in ontology mapping and using very complicated data sets, and particularly for standardized data that can be used for multiple purposes. So would you elaborate on where you think the utility is and what gets you excited about this just massive data warehouse?
Sure, Collin, thanks. So we've already heard a lot in the past day and a half about the importance of getting the most value from every data point in rare diseases. And one way that we do that is by combining data from different sources and from different data sets, allowing us to make larger cohorts for more powerful analyses, to make connections across different types of data. And I think that's where RDCA DAP and CPATH are really an important partner in this partnership, the public-private partnership. We have many years of experience in curating and standardizing data to different models, from CDISCs, standard study data tabulation model, the OMOP common data model, OBO ontologies, which are very semantically rich ontologies that can combine things with sources like the OMOP standard vocabularies. So being able to, and using those models on different data types, right? So we have a lot of experience working with clinical data, working with DHT, digital health technology data, working with registry data. And so combining those kinds of more traditional data with some of the newer biospecimens that are being collected as part of all ALS, I think is where we're going to really extract the values there. And I think CPATH's experience and RDCA DAP's experience as a trusted partner in this field has allowed us to really, and will continue to allow us to expand the collaboration to include more partners from industry, more patient organizations, a broader voice, and really bring all of those in there. And then, of course, you mentioned about ontologies and knowledge graphs, right? So the standardization of this data, particularly when you've got a situation like this where data are being collected by different partners, being able to speak a common language across all of those different data sources is what's going to allow us to combine them to get the most value from it. And so we're really excited to bring AMP-ALS and related neurodegenerative disorders into our knowledge graph so that we can start to inform things like, well, first of all, what is ALS, right? It's not a uniform disease. So how can we really start to understand that using the bigger picture of all of that data? can we inform ALS research using knowledge that's been gained from more common neurological diseases? Can we use ALS data that the knowledge that we gain from this project to inform even more rare neurological disorders that we have data about? So the knowledge graphs allows us to ask questions like, okay, so find me the most common phenotypes in the patients in this data set, then go out and discover from public sources what genotypes are already associated with those. Then what data are connected? What do we know about the proteins or expression levels of those different data sets? How can we link that back to other diseases, et cetera, right? So being able to do those very sophisticated queries that span many data sets, I think is a big value of what we're building within CPAT.
I'm going to switch to Teresa, and Teresa, I mean, obviously you spent a lot of time in this space. The FDA has been very supportive of the COA discussions, and even providing insight with us to all ALS. I mean, what are your thoughts about your experience with PPPs? Obviously, you've been involved in many of them, and what are some of your hopes for what we're doing, and steps that you'd like to see move forward?
So we're very engaged in the efforts both with CPR&D and AMP-ALS, and I'm really excited about the work that it's just starting. We're just getting going, and we've got a lot of exciting initiatives and projects underway. One of the things that we've been working on, as you mentioned, was with COAs, where we've been working on trying to I think the first step has been to try to quantify or describe what the existing clinical outcome assessments that are out there are, and better understand their utility and where there are gaps and needs for new and improved clinical outcome assessments. And so that's, I think, a critical piece in order to understand what clinical benefit we're seeing with these therapies are. But aside from that, looking at more commonalities across diseases, each disease is unique. However, there can be commonalities, particularly when we're dealing with neurodegenerative diseases, where the specific etiologies of the diseases differ, but once neurodegeneration is underway, many of the same neurodegenerative processes play out across diseases. So biomarkers that are relevant in one disease may very well be relevant to other neurodegenerative diseases. Symptoms may, as I said, each disease is unique, but there can be symptom overlaps. So even if we are developing a clinical outcome assessment in one disease, it very well may be able to be leveraged, or at least pieces of it may be leveraged across diseases. Natural history studies are common to all of these diseases. We need to understand natural history data. So there's a lot of shared learning that can be shared across the diseases, and how to optimize the design and data collection and natural history studies, how to make not only in the design, but how to, decentralize trials, as Amy was mentioning, being able to have remote assessments and understand what can we do remotely. We understand that coming into clinical trial sites is really burdensome for patients. And I think that we would greatly increase both the number of patients and the diversity of patients that we enroll if we are able to have more trials that are decentralized and able to be done either in the home or in local sites. So, these are all things that I think are common across the disease areas, and having initiatives like CPR&D, where different diseases are sort of coming together under a single framework, really allows us to share those learnings across diseases. It makes things more efficient overall, and decreases the amount of resources any individual group has to put into the programs.
Thank you.
Yeah.
Brian, would you like to comment? I mean, you've been involved, and your company's been involved in some of these discussions. Like for industry, like what gets you excited? What value do you have in this? Obviously, anytime you're working in rare disease, it's already a complicated space to get into. But from what you've heard, and what gets you excited, and why is this valuable to the pharmaceutical industry?
So I've been in industry for almost 35 years in clinical development and regulatory affairs, most recently in the biotech. And what I can say is that I'll speak to some of the challenges we have is that in biotech, we're focused on execution and innovation. And in doing so, it's no surprise that over the past several years, about 1,000 biotechs have been part of mergers and acquisitions a year. So the big pharma has the pockets, the biotechs have investors that we're looking at. So one of the challenges on participating in programs like this is we need to look at where every dollar goes. And we need to have a return on the investment. So we don't have big pockets to pull from to participate. And that may, provide some or may lead to some bias in the voices that you actually hear. So we need to be careful of that. The second thing is from a personnel perspective, a challenge is I'm an N of 1 within our company. I'm working on regulatory responses that are due this week. I've got other activities to manage. I'm sitting here with FDAers that I see at a lot of these meetings. I don't know how you do it. You've got 61 of these programs that at least I saw on your website. These programs you're doing. You have reviews that you're doing. I heard yesterday, Dr. Campbell, you're working on a cloning device. I hope that you get that. We all can retire or work or both. So that's a challenge. It's a big challenge for all of us, our resources. But I want to say that one of the other challenges, and we're always talking about data sharing. Why are we talking about data sharing? Because companies are filled with egos, careers, and lawyers. So we need to get away from that. This data sharing, we're all, I'm preaching to the choir here. We all need to data share, and there are a lot of examples of where that is successful. We need to get to it. I like yesterday I saw a diagram, and we've talked about this, Collin, is that having CPATH in the middle and having data in different areas that are controlled by the companies, yet there's a commonality that you can bring it together and work on tools and other things. I think that's a great selling point. We just need to get that word out within our own companies. So those are some of the challenges. I love working. Some of the benefits might be I love working with these people. I'm here today because we've got big challenges to overcome, and I'm mainly working in ALS. In a short period of time, these patients aren't going to live. Many of them aren't going to live past the clinical trial data readout, yet alone a therapy that is meaningful. So we have a collective mindset that we're looking at, and being able to come together and tackle these large problems in a short period of time, I think is a really strong benefit of these public-private partnerships. We get a voice in what is worked on. We hear a lot about biomarkers. That's a short-term need, especially in these rare diseases. A longer-term need is working on the underlying mechanism of the diseases. That's longer-term, and in biotech, yes, we're looking at that, but that's over the horizon. And so I'm hoping some of these databases can actually get to some of these mechanisms, because it's the mechanisms, once they're identified, can allow academia and biotech to work on actually making a drug that can work on these targets. But I think the biggest benefit in my mind is the being able to sit up here with the agency and understand their insights, understand their issues, understand their priorities. And I think it goes vice versa. I think it goes the other way too. They understand what we're facing. I think that's one of the biggest benefits of these partnerships. And in saying that, I think also having a public forum dispel some of the misconceptions that the public has on collusion or unfair cooperation, or we're working together against an evil In my 35 years, I haven't seen it with any of the Western agencies. So I think it's been very collaborative, and I've seen a lot of, and I'm fortunate to work in an area of ALS where there's a lot of work with the FDA in trying to accelerate therapies.
Before you go to Olga, I mean, do you want to comment to something that Brian said with you talked about a little bit on ontologies and how understanding mechanisms of disease and how this data warehouse might help with that?
Oh yeah, I think that's
Yeah.
that's really key to having the data warehouse. I like, the fact that the last session was entitled From Data to Knowledge. I was like, why am I not talking in that one? Because that's what we're trying to do, is transform that data into knowledge and using existing human knowledge to do that, right? So there's a lot of buzz about AI right now, and AI is going to help us. It's going to be game changing, but using it well is very challenging. And so having very well curated data where all of the steps are traceable and all the changes that were made to that data before you analyze it is crucial. But also just asking the questions. Often we know that there's information out there to discover, but we're not sure what that information is, or even if we know exactly what kind of data we want. When you're searching across a data warehouse that has 10 different kinds of data, you may not know the technical query that you need to get to that piece of information. And so large language models, big buzzword right now, and there's a reason for them. They're transforming the way that we can query across data, but everyone I'm sure knows about hallucinations, right? So how do we get rid of hallucinations? Ontologies. Ontologies, you talk about the human in the loop. Ontologies are the human coded into a machine in the loop, right? So ontologies are a machine and understandable representation of human knowledge. And when you combine ontologies with large language models into something called retrieval augmented generation, that's what allows you to get answers that are not hallucinations, but also to discover, they tell you how to ask your questions so that you can get the information that you want. And I think that's really crucial.
Okay. Olga, same question that I asked, Brian. I mean, as you're earlier on in development in, neurodegenerative diseases, and, there's obviously you've sort of probably done the landscape of the challenges ahead and whatnot. I mean, initiatives like this, what hope do they offer you, and what are your thoughts about the outcomes and hopes for those solutions?
So thank you for letting me participate today. It's really a pleasure. My name is Olga Björklund, and I work in a small biotech company in Sweden called BioArctic. Now, we do have a bit of a unique perspective on things because lecanemab was discovered by Professor Lamfeld, who is grounded the company. And for many years, we have followed the work in Alzheimer's disease, and all the work, the clinical path, really brought through and helped the clinical trials be formed so that even lecanemab had a chance to really show its benefit, and ASI took it really in a smart way through the clinical development. So we were very excited to understand, because we are a company who have monoclonal antibodies for Alzheimer's disease, for aggregates in Parkinson's disease, and now for TDP-43 aggregates in ALS. To see that the path for ALS has been created, and we are really, it didn't take long to persuade our leadership of the importance of the C path. So as a small biotech, we see that this is a sort of a one-stop shop. We can go to you and ask about, okay, we are in the pre-IND stage. What is going on? What are the key questions? What are companies asking? What are agencies replying? What are their views? Because it's not like we can really achieve to go on many early meetings within Europe, within US. So that's really the knowledge that we're seeking for, and why I see really the big positive thing. Then a small biotech, and then just to build on a bit what Brian said, I mean, the resources are limited. As a regulatory lead at BioArctic, I mean, I'm contacted, and my clinical colleagues are contacted all the time by different DHT companies, and then we would like to put in everything in our trials. Then again, it's not possible. So we really need to dissect what is good, what the patients want to see, how not to make these clinical trials so burdensome, but still get the information we need to get to the next milestone. And this is really a great repository and the way to monitor and which are really the good and prospective DHDs, clinical outcomes that can be sought. Then the biomarkers, I mean, for us, it's really crucial to know what we can build on, how early in the development we can find the relevant biomarkers. Perhaps not all the biomarkers that have been out there are relevant for our mechanism of actions. There is a plethora, but then they need to be validated. So all that really needs to be put in place. So that's why I see the CPATH and these fantastic initiatives by NIH and ALL as really good.
Thank you. Thank you. I'm going to ask Teresa, Amy, and Shubhangi to comment on one thing. I mean, obviously, I talked in the intro slide about like learning from each other. I mean, and the efficiencies of that. Do you want to talk to that point and how you've observed within the discussions that we've had and how this can help this model, you know, with looking at data and, you know, identifying gaps and then fulfilling the gaps, you know, where do you see that in this initiative, and you know what kind of examples you might have in from already our conversations? And do you want to go first?
Sure, I'll start. So I will say that, you know, we've talked about from industry perspective of limited resources and more efficiency. It also applies to FDA as well. I mean, we have limited resources as well. And so there is efficiency for us in order to, in participating, and we do find value in participating in these consortia, because that does give us an opportunity to share our thoughts broadly outside of the context of any individual program. And hopefully it will, you know, ultimately might reduce the number of meetings that companies might need to request if they can get some common knowledge that they can take back. You know, we put out guidances, but those are a lot of work for us. So having these sort of informal conversations through the consortia are really helpful for us as well, to be able to share our thinking in a less formal way than an official guidance document. I'll see if Amy or Shubhangi have anything.
Amy, do you want to go first? I saw you come off mic.
Yeah, sure. So it's this is a great conversation, and I really just want to say that, you know, it's to echo what other folks have been saying about limited resources. You know, it's been quite a lot of exciting work, but a lot of work to really get the All ALS Clinical Consortium in place. And I think in terms of collaboration, it's just been really fabulous to be able to have a conversation as we were developing the All ALS protocol last winter with FDA and CPR&D around some of the clinical outcome assessments that were being considered. And, you know, as it happens, you know, the insights that we received were really great, but also some of the patient-reported outcomes that All ALS is planning to collect, then I think we're integrated into this landscape analysis that's being performed. And, you know, we are looking forward to insights from that. And I think that's just a really tangible example of how it's been great to be able to have this these sorts of ability to have a conversation as things are being put into place. And, you know, depending on, you know, the results of that report, you know, we're really hopeful that All ALS is agile enough to be able to gather additional data as needed to be able to support, you know, insights that will be provided from that report about the need for data on validation.
Thank you. Shubhangi?
Oh, so as I described earlier, we have several ALS objectives, and we have various groups working together, contributing to a more comprehensive approach in fighting ALS. It's important to remember that these initiatives, all of them work in synergy. They are not individual efforts. So for example, the knowledge platform that we are building will bring in the data sets that, you know, already exist, will bring in the new data sets from the natural history study, and which will allow researchers to share data across, you know, and then can inform biomarker development, ultimately leading to better diagnosis and treatment efforts. This collaborative approach is what will make AMP-ALS so powerful. So collaboration between public and private institutions is crucial in building data. By sharing data and expertise, public and private institutions can accelerate progress. Centralized data sets are again a game changer. Researchers can analyze vast amounts of data from these various resources. All that will exist in one place and can lead to faster breakthroughs in understanding ALS and maybe identifying new treatment targets. So yeah, this will give them a much bigger picture and is a powerful tool with the potential to accelerate research in this space.
Thank you.
Thank
I'm going to
you.
add to that as another example. The biomarker working group has originated into the AMP program. Obviously, they're looking at new biomarker development. But in parallel, we're actually taking existing data to identify gaps within the data to define what the context of use and where data might be needed to supplement research to get a biomarker endorsed through the qualification or through a letter of support. So this interplay and this conversations are honestly, they're going all of the time. The same thing as the new biomarker experiments discussions are going on. There's discussions about how the design might affect the context of use or the regulatory strategy. And so these are happening in an incubated system versus you have to cook the data and it comes and then you decide if it's good for regulatory purposes or not. In real time, this interactive play has been really, really important. I'll pass it to Alex to finish questions. And I'm
So
going to say,
thanks, Collin. Well, I came to the panel with 2 questions, but you guys already answered one. Thank you. But just getting back to the question and turning that into a comment. The question was about CPR&D, and you described it's not just for ALS, right? There's a decision to call it rare neurodegenerative disease because there is a broader intent, not just to help ALS, but to help rare neurodegenerative disease. So the question was about how do you think the work for CPR&D is going to help inform and accelerate efforts for the rare neurodegenerative disease? So Teresa and Ramona, you already provided some good answers to that. I wonder if any other panelists had some additional comments. Olga? Yeah.
Well, I think for me, it was fantastic to learn about this initiative and what you are doing, because there are so many data sets and when starting a project, we are looking at where should we go into, which one is the most reliable, what has been measured, what can we extrapolate from it. And it's there are so many academic research and these registries, and just to understand that there is a standardized way that we can pull the data from and see what fits our program and ask the relevant questions and then get into the modeling. That for me that is such a powerful tool because the standardized data will really help us to understand our drugs and development. So
Great,
I'm
thank
thankful.
I'd like to answer that with 2 ways. Number one is learning from these other programs that it's worth looking at. The other thing is that and I'm not a neurologist, I'm a pharmacist, but neurodegenerative diseases, there's a lot of overlap in some of these diseases. And I know at our company, we're looking at some of the same drugs working in different disease states and different populations. So it's not just ALS that we're working on, and it's neurodegenerative is a blanket, but there's so much interplay between the neurodegenerative diseases that we recognize, and I think the FDA recognizes too.
I'd like to speak to a specific example, which we touched on in the prior session, which was Hovinga, and how that that is a great example, I think, for how we were able to leverage some of the learnings that some of the efforts that CPRND is involved in, but that there's learning there for across diseases as well, even though so that was Juvenistat was for Duchenne muscular dystrophy, and it was approved earlier this year for patients 6 years and older. And it was a rather challenging review for us. Of course, Duchenne is a rare disease, and it's a terrible progressive disease. And this was a situation where we had a single adequate and well-controlled study, and we were able to use confirmatory evidence to support a substantial evidence of effectiveness. And this is becoming an important pathway for us in rare diseases. This one positive study plus confirmatory evidence, because we understand that in rare diseases, it can be really challenging to do 2 studies, which would be our usual standard in more prevalent diseases. And so confirmatory evidence is becoming more and more important to us, and how we sort of make a case for confirmatory evidence. Now, last year when we were at this meeting, we had approved omaveloxolone for Friedreich's ataxia. And so there was a lot of discussion at that meeting about how we use natural history studies as confirmatory evidence for that disease. And now I think the Juvenistat is sort of the following story that follows on what we've done with Friedreich's ataxia. And first, with the single study itself, there were some challenges with that study and in the dosing in that study, which I don't think we touched on earlier, but there was a dose escalation, but there were also some safety issues with doses that required dosing adjustment. That made it challenging to interpret the data and help us inform which doses to recommend. So we did use modeling for exposure response. So modeling was very important to that application. And I know that's one of the specialties of CPATH, is your modeling work. That also informed both the exposure response and then ultimately how we described dosing bands in the labeling. But then the confirmatory evidence in that study was also very important. We were able to use 2 main pieces of confirmatory evidence. One was natural history studies in Duchenne that we were able to help us and confirm the findings that were seen in the single study using imaging DMD and synergy data sets. And then the other piece of that was in our ability to use MRI, fat fraction MRI from the vastus lateralis. And I know that's something that in the Duchenne consortium is working on, I believe, a clinical trial simulation tool using fat fraction and MRI. And this is the first time we've had it as a regulatory use for this disease. We've always thought it seemed very promising. We've been very encouraging of the work that DRSC has done in this space, but we haven't had enough data to really use it as a primary or even a secondary endpoint. And so this was the first time that we've been able to have some data that we were able to leverage together as confirmatory evidence. And I think that there is, the pieces that I see to learn from that are, the importance of modeling for exposure response and to inform dosing, the CE, the ability to leverage fat fraction MRI, which would not necessarily be limited to Duchenne. There may be other neuromuscular diseases that could be useful for. And again, similar to what we had with Friedreich's ataxia was the natural history studies. So I see that as a great success story for that approval, that I think there's a lot to learn from that. And hopefully people will go in and look at, our reviews are posted publicly. You're welcome, you can go and look at our reviews and see our thinking on that application.
Great, thank you, Teresa. And since you mentioned Felix, I put a plug in for myself, but we have the RDCA data is not just a platform, it's also a program, and we're actively leveraging the data that is flowing, so CPRND, but also through simply the RDCA data platform. So we do have active work and modeling efforts in Friedreich's. It's taking off in progressive supranuclear palsy as well. So just to mention that internally, as a collaborative effort between RDCA DAP and CPRND, we're also working together to try to accelerate efforts, leveraging the data, and hopefully some of these modeling efforts will help, and we'll be able to extract lessons learned to inform CPRND activities.
I just, something that's not currently part of the AMP ALS program, but I think that's CPAS rare and orphan diseases programs have developed quite a bit of insight into, is using real world data from electronic health records. that's another area where there's a lot of buzz right now, but I don't think very many people understand just how difficult it is to get good data from EHR records. And we're fortunate that we have, through other programs, been able to build a very robust real world data ingestion and curation pipeline. Again, one of a handful in the world that are integrating multiple EHR data sets using a common data model. And I think the lessons that we learned from that, and that we can through, our neonatal consortium, through the RDCA DAP, we failed early and very well with the FA real world data effort that we experimented with. But we learned a lot about what you need to do to be able to work with real world data. And, I think there's great potential to apply those learnings to AMP-ALS, to rare neurodegenerative diseases, to understanding that natural history for use in that confirmatory evidence from real world data. As there's a really big potential there.
Right. Thank you, Ramona.
Bob, do you have any others?
Yes, I have one final question. If you
Go.
if you please. Now that you're sitting down.
Yeah,
Yeah, no, just a final question. So the connect the dots question, we've talked about different pieces of this very encompassing framework that is the CPR&D. And you're all coming from different part of these pieces. So taking everything high level, how or why do you think this framework is going to help accelerate drug developments for ALS? Brian?
So coming from the sponsor perspective, and we've heard about this, and we heard Dr. Cabezzoni talk about regulatory framework. We need to work together on how this all comes together, because ultimately, whatever we think it works may not satisfy the agencies. But I'd like to see the bringing together the real world evidence, real world data, the EHTs, the biomarkers. I'd like to bring it to where we have a regulatory framework that satisfies the FDA and other agencies, where we're just doing one trial. We just do one trial that can be adapted and move forward. And at some point, you either fail or you succeed. Not every trial that we design, even for a similar disease state, is the same. I have a pleasure of working with Dr. Genge, and we did a 3+3 design for ALS up in Canada at one point, taking the oncology paradigm. It was very successful. We're working on another drug where we're doing similar drugs. We're doing two different designs from the clinical perspective. So that's what makes my job interesting. We are looking at how we can innovate in clinical designs to accelerate drug development, and using these databases, using this type of forum helps us get there. But I like to see one trial eventually. No placebo.
Thank you.
I can follow from that. I mean, I think AMP-ALS in a way is a realization of a dream that many of us have about getting all the data at scale to get a full picture of the populations, the individuals, the disease itself. It's a real rare opportunity to have this level of funding and this level of collaboration. And so we need it to succeed, because the government that actually decided that had the courage to fund this effort, if we can make this work, then it can be used as a model for other disease areas and expanding again to more rare neurological diseases more generally. And so that's what really excites me about this program.
Thank you. Maybe Amy or Shubhangi, do you have a comment? Amy?
Yeah, sure. I mean, I think I would just echo some of the things that have been said. I mean, it is, as I have said to our PIs, sort of a once in a lifetime opportunity that we have here, where all the stars have aligned to give us this opportunity to collect these data at scale. And, our hope is that, the structures that have been put into place to make the data available through the knowledge portal, and also specimens being collected in an NIH biorepository. These are measures that will hopefully allow researchers and the public access to these data. And we're really hopeful that will support collaborations that will accelerate cures for ALS.
Thank you. Shubhangi?
Yeah, I don't have anything additional to add, but I want to re-emphasize what other panelists have said. I think we are at a very exciting time. We have a lot of great work going on, multiple efforts in this space, and all of them are coming together and will hopefully bear fruit in the next few years through AMP-ALS. We are hoping to share data broadly and quickly with the field. So this will raise the entire field and not just a few individual stakeholders. So there is going to be a benefit to all the stakeholders and will get the field, get the field to move in a direction that will get us closer, hopefully, to a cure.
Right,
Thank you.
Any additional comments, Collin, maybe, or
No.
do we go for questions?
We'll go for questions, the audience.
Yeah. So we'll take questions from the audience.
So Amy, this question is for you. I was struck by your introduction and how you guys are laying out the all ALS program. And I want to ask a favor, which is, it doesn't matter if you get all 2,000 or if you don't. The approach that you guys are using is so unique that that lesson, good or bad, would be really powerful for sponsors who are trying to reach groups who are not being reached. So what I'd like to ask is if you will publish on the results. It doesn't matter if it's good or bad, because you'll be able to say what worked, what didn't, maybe what you could do differently. And so I don't care if it doesn't all work, because it's all going to be important for us.
Thanks so much for that comment and that encouragement. We are really excited about this approach, and I think we have started enrolling, but I think we don't know how it's all going to pan out. We're very hopeful. We've been intentional with the goal of trying to capture really all populations. And I do think, as is consistent with the spirit of AMP-ALS, those findings, we will certainly share those. I know there's a lot of interest I've heard in, the feasibility of remote enrollment on this scale. So I can certainly make that promise to you that we will disseminate those findings and hope that they will be used.
Great, thank you, Amy. See your question here?
Yeah, quick question. Colin from the ALS Association. Could you describe for the all ALS study, what's the plan for data storage and data analysis? And the reason why I ask is, one of the issues we're seeing is where NIH has funded large consortia to collect data and collect samples. And then when the grant ends, the funding for storage and sample collection ends. And PIs are now asking the association for funding for, keeping their samples, because it takes up space in freezers. The terabytes and terabytes data that needs to be hosted on some kind of data portal takes up money, but that funding is never there once NIH funding runs out. So in all ALS study, how are we going to make sure that after the 5 years of funding is done, that funding for storage and then funding for analysis, so incentivizing folks to go in and either take those samples or take the data, how is that going to work?
So thanks, Colin, for the question. I wish that we could be more certain about the funding over the next couple of years. So it's a good question to ask. Perhaps I'll defer to, I don't know, maybe perhaps Shubhangi can speak a little bit to the knowledge portal question, but I can speak to the biospecimen question. I mean, so the samples that All ALS collects are going to be housed at BioSend, and that's an NIH repository. And that funding is not, it comes from a different stream, and I don't anticipate that being, those samples being at risk, you know, in the future. I think we are trying to, I think if there are other studies that have samples that could be used as part of this ALS effort, I think be happy to, you know, speak with you or other researchers about, you know, even bringing those into the biorepository. That is, I think, one of NIH's goals, to really create, you know, a large collection in that repository that researchers can access in the future.
I can add, you know, a little bit about the data portal. So the knowledge platforms or the portals that we are building as part of AMP-ALS, they are actually a part of a lot more, of a few more AMP programs. So we have an AD portal and a PD portal, and I know that we are having discussions with NIH for the long-term sustainability of these platforms. These are not going to go away, from my understanding, but we are figuring out a plan for a long-term sustainability, because we want to ensure that all the great amount of data that we are putting together as part of all of these AMP programs can exist in perpetuity and can be used by the researchers. Thank you, Kuldeep. We can always connect later on and discuss this more.
Thank you. I don't see any other questions. So with that said, I want to thank the panelists for a very interesting discussion.